career as a physician. And there was a different picture that I wanted, but I found this one, and I thought that was probably an even better one to start with. Because I got right around then, this was his bar mitzvah, as I understand, he was already plotting his career as a physician. So he really had that perspective through his entire career, but in the late 70s and early 80s, when I first knew him, he was wont to say the MD after his name stood for mouse doctor. And I remember very vividly him reapplying for his New York license for one purpose, and that was to write antibiotic prescriptions for Adam, who was getting ear infections, and he didn't want to deal with a pediatrician. So that was right around the time of the Citizenship Party, if I recall correctly. In the last decades, though, as dendritic cell science and Ralph's career progressed, Ralph's interests came full circle. So this is the time when he was a mouse doctor. So there we go. His interests came full circle, and he became very interested in moving his discoveries into the clinic and harnessing the power of dendritic cells to address important medical problems. Of course, while doing this, he continued his first great passion, elucidating the basic science of dendritic cells, or as he often described it, learning the rules of immunology. As those of you who engage in human clinical investigation know, it is a work fraught with bureaucracy, bureaucrats, paperwork, and delays. It requires much patience. In the end, much of this is in the interest of protecting human subjects. However, as those of you who knew Ralph know, this was not a match made in heaven for Ralph, with his passion, urgency, and frankly lack of patience. However, Ralph entered the fray with his usual enthusiasm and vigor because he was so convinced that doing this type of science was absolutely critical to solving the medically significant problems that he believed dendritic cells could address. In the mid-90s, Ralph, together with Nina Bardwaj and Madhav Dadapkar, Ralph did the first human clinical trials of making dendritic cells with blood cursors. They used the newly available recombinant human cytokines to drive them to become dendritic cells, and then re-injected them into humans. By doing this, they demonstrated that using this method, these dendritic cells could induce immunity to a new antigen, KLH, and also recall antigens, flu and tetanus. This provided the basis of all of the many clinical trials that are now moving forward using variations on this technology to treat cancer. We'll hear about this work or other work like this from Madhav, as well as from Carolina when they speak in this session. I was at the Rare in Washington at this time, and I came up here to learn the technology so we could use it to demonstrate that dendritic cells could induce immunity in humans when loaded with the Rare's canary pox-based HIV vaccine. This is the vaccine that was later employed in the Thai trial that demonstrated that humans could in fact be protected from HIV with the vaccine. Ralph brought me into his office and sternly warned me not to get too invested in this technology. I was still shocked we could do it at all. He explained that though it was very new, it was already outmoded in his mind. I think this was 1996 or 1997, and he was very clear that we would not be taking out precursors and making DCs to reinfuse for the long term. Once the principle was proven, he said, we would find dendritic cells where we live and target them in situ. Needless to say, I was impressed, but not really surprised. The last thing that I thought was I would spend nearly a decade of my life working on this with him. As we understand now, Ralph was referring to targeting dendritic cells through DEC 205 and for vaccines for infectious diseases and cancer. Since 2004, with funding from the Bill and Melinda Gates Foundation, much of the work in Ralph's lab has been directed to bringing this into humans. My friends and very close colleagues, Christine Trumfeller and Marina Kasky, will tell you about this work now in the first talk of this session. But before I turn over the podium, I just want to close my very small remarks with this quote from Ralph. So at some point in one of the last years, I wrote to him and asked him for the quote that many of you have seen shown, that he liked to show, about chance favoring the compared mind by Louis Pasteur. And he must have been inspired because he sent me a list of about 6 or 8 quotes that he liked. And he ended with this, which I had never heard him say, but this was his own attribution. And he said, being explicit about patient-based research, it is exasperating in the number of significant obstacles that must be overcome, essential for better understanding of disease development and treatment, and equally profound to other spheres of scientific research. Excited. So with that, Christine? Christine Trumfeller is a research associate in Ralph's lab and our director of preclinical activities. She came to us from Klaus Huberlau's lab and has been working here at the Rockefeller for a decade. And I'm going to introduce Marina because she's going to come following Christine, and I'm not going to get up again. And Marina is an assistant professor of clinical investigation in Ralph's lab, and she was a graduate of our clinical scholars program and trained in infectious diseases at Cornell. And as Ralph liked to say on a regular basis, she's a real doctor.
Thanks, Sally, and thanks also to Kai, Michelle, and Ira for inviting me to this symposium on Ralph. And as Sally already mentioned, one question that Ralph wanted to answer was whether dendritic cells can be harnessed to design vaccines, particularly vaccines that are based on T-cell immunity for infectious diseases like HIV or also for cancer. And basically everyone in the lab was working on this project over the last couple of years, and we're going to present some of the work here today. And we're going to split up the presentation in two parts. I will tell you a little bit about the rationale and the preclinical evaluation of this targeted HIV-gag-protected protein vaccine. And then Marina is going to talk about our efforts to bring the vaccine into the clinic, and will give you a short update on our ongoing phase 1 study in humans. So Ralph was inspired by Pasteur, and as you know, Pasteur was a microbiologist who developed vaccines activating or attenuating microbes. And up to now, vaccines have been developed by these principles, with examples being, for instance, rabies, yellow fever, and polio. However, this approach could not be or wouldn't be safe to use in terms of an HIV vaccine, and also cannot be just extended for cancer. So what Ralph had in mind was to explore an immunological approach that emphasizes dendritic cells, obviously. So here the vaccines would be comprised of defined protective protein antigens that are given with a defined maturation stimulus or adjuvants, and these vaccines would be delivered directly to large numbers of dendritic cells in situ. So protein vaccines are safe, and normally they are also less expensive to produce than other types of vaccines. However, they are also sometimes purely immunogenic in terms of T-cell responses, and even when given at high doses. To improve the immunogenicity of protein vaccines, one can exploit several features of dendritic cells. For example, dendritic cells express receptors for antigen uptake and processing, and they also, like many other cell types, express several pattern recognition receptors, and signaling through these receptors lead to the full maturation of dendritic cells. So, dendritic cells are heterogeneous and can be divided in several subsets, as we already heard many times today. For example, in a spleen of a mouse, you can find the CD8 positive and the CD8 negative dendritic subset, as well as plasmacytoid dendritic cells. Each subset is characterized by a distinct expression of uptake receptors. For instance, on the CD8 positive disease, you find declin-5, TRML4, CLEC9A, and langerin, while on the CD8 negative dendritic cells, you find TCR2. Also, these subsets express distinct pattern recognition receptors, like, for instance, the TLR3 and MDA5 on the CD8 positive subset. And as you know, we picked as our first choice of antigen uptake receptor DEC25, and we also worked with poly-IC as our first choice of adjuvant. Also, as already told this morning, poly-IC is a synthetic double-stranded RNA molecule that signals through MDA5 and TLR3. So the strategy we have been using is to target vaccines to dendritic cells in situ by engineering vaccine proteins into anti-DEC25 monoclonal antibodies. And this strategy was first worked out by Daniel Helwiger, who was a student in Michelle's lab.